Okay, we're recording now. All right, start over. So everything's made up of carbon. Your cells are made up of organelles, which are made up of molecules, which in us is mostly made up of carbon. You may know what this is? Plant cell. What's this? Animal cell. Eukaryote or prokaryote?
Which one? Oh, wait. I'm sorry. Eukaryote. I knew that. I'm sorry. I'll state it.
Eukaryote. We'll get to that again later on.
Sorry, that was stupid. I'm so sorry.
This is stupid.
I knew that. No, that was too stupid. I think I forgot
I'm
that.
alive.
It's terrible. Wow, it's so bad now.
How come if you listen carbon is 70% water?
Well, of the compounds that you're made up of that are carbon based, they're carbon based. But I mean, you're mostly water. But of the molecules that make you up besides water, they're carbon based, all of them. That makes sense?
Yeah.
Yeah. I mean, I see what you're saying, but. So we're mostly water, like your whole body, humans, your whole body's around 60-something%, depending on what part of your body you're talking about, what organs, your different percentages. Anyway, it's about 25% carbon compounds, proteins, carbohydrates, nucleic acids, and lipids, which is all what chapter 5 is on. And so we'll get to these a little later. You should remember some of those from Bio 1. Probably talking a lot more detail in here about them. So, about 3% salt. Salts are important for maintaining what we call osmotic balance. If you ever go to the hospital and get an IV, they don't just give you distilled water because they would completely mess up your osmotic balance. They give you a saline solution that has salts in it. It's an isotonic solution, if you remember that, what that word means. We'll talk about that in chapter 7. So your book talks about the synthesis of organic compounds. So early Earth, like if I transported you back in time 4 billion years ago, I would kill you, so I wouldn't do that. But early Earth, there was no atmosphere at all, like no atmosphere. The Earth was constantly bombarded with like UV radiation. And you don't really think about how much our atmosphere protects you from UV radiation, but it's a huge amount of protection. If you took away the atmosphere, you'd go outside and get burned within like 2 minutes. It's really, really fast. So there's a lot of protection there. But because there was an atmosphere, everything on Earth was being UV irradiated a whole lot. There was lots of lightning and storms. And we were what we call a very reducing atmosphere. There was no oxygen, so you would suffocate, and no organic compounds. So the main components would be methane gas, hydrogen gas. There was carbon dioxide. We don't say carbon dioxide is an organic compound per se because of the gas. When we talk about organic compounds, we're talking about like carbohydrates and stuff like that. Anyway, so there wasn't a whole lot there. There was definitely no life at all. And that's like, anyway, so a guy named Stanley Miller, a long time ago at the University of Chicago, I think in the 50s, he showed in his lab the synthesis of organic compounds using inorganic substances. And he basically used this giant still apparatus. This looks actually pretty small. His actual setup was giant. I think there's a picture in your book somewhere. I'm not sure if it's in this chapter, but it was like a whole like giant lab bench. I mean, all these giant glass tubes, and it was huge. Anyway, what he did is up here, he created this sort of atmosphere. So methane gas, ammonia, hydrogen gas. He had electrodes which would simulate the lightning. And then there was water on early Earth. And he simulated water vapor by heating up water. And then here we had water vapor go over here. And it would pass through this quote-unquote atmosphere area. And then he had a condenser where the water would condense into liquid form. And then he collected it down here. And he analyzed it. And he actually showed that he synthesized basic organic compounds like amino acids. He showed, so what this is called is abiotic synthesis. So synthesis of from inorganic compounds of organic compounds. And it basically just showed that it could happen. One of the things we'll talk about later in this class is like, how did life come to be? Like from non-living, where did life come from? And to be honest, no one actually knows. There are no experiments that have shown the synthesis of a cell from like nothing, a whole cell. There's been experiments that have shown that organic compounds can be synthesized from inorganic substances, that something cell-like could form that has basic metabolism, and we'll talk about this later on, chapter 25. But no one knows. So anyway, that's all this was for now. Let's move on. I'll get to all this stuff later. Organic chemistry is the study of carbon-containing compounds. And if you go to college and you decide you want to go into medicine, or you're a biology major, or even if you're a chemistry major, you're going to take organic chemistry. Like it's a whole class in college, like a whole year worth. Some people love it, some people hate it. I was one of the people that didn't enjoy organic chemistry. My husband loved it. Anyway, he's a chemistry nerd. But this is basic organic chemistry we're talking about in here, and this is the stuff I like. Anyway, all right, so carbon-containing compounds. Carbon, the answer to the first question on that note was the reason why carbon is, well, second question. The reason why carbon is so interesting is because it has something called tetravalence. It has 4 electrons in its valence. How many more does it need in order to be happy? 4 more. So it's not going to like give away its 4. It's not going to take 4 more, so it's not going to participate in ionic bonds. It has a tendency to participate in covalent bonds. And it can form or will form 4 covalent bonds called tetravalent. Because of those 4 electrons in its valence shell. It'll form 4 covalent bonds. So it can form these super complex molecules because it can make all these different bonds. And this is a very simple organic compound, CH4 methane gas. And carbon can never form more than 4 bonds. It will not. If you ever see something with a 5th bond coming off of carbon, like that's incorrect. That's not, you can never do that. It can form double bonds and triple bonds, but it can only have 4 total bonds. So if carbon is participating in, it can do that. What is that? CO2. But anyway, so it has 4 bonds. So 1, 2, 3, 4. See that? You can't form any more. There's only 4. This is a very simple, what we call hydrocarbon. A hydrocarbon is just a compound that has carbons and hydrogens. All of those bonds in there are nonpolar covalent bonds. So this molecule is actually gas at room temperature, but it would not associate with water. So, okay, I hope you don't know me yet. You will. Please don't take this offensively. I just think it's really funny. Sorry. But look, look at the one. See, 1, 2, 3, 4, 1, 2, 3, 4. 4. Sorry, it's, yeah. Anyway, I said that before last time. I said, no, chlorine is a 4. I was like, this is in chemistry. Sorry. Never mind. When you Google image stuff, you get really weird things, and that was one of the things I got yesterday when I was Googling imaging carbon chemistry. This is a picture in your book. These are all different hydrocarbons. Notice, you don't need to know about the geometry. If you took chemistry, you learned about the geometry and the angles of bonds and all that crazy stuff. You don't need to know that for this class. I just want to mention it. I will tell you, however, you do need to understand that the number of bonds will depend, well, like the 1 bond versus 2, this is a much stronger association than double bond. And having 3 bonds is even stronger. But also, in this particular configuration, this is ethane gas. But the way, do you remember using those like molecule kits last year where you could move them around? Yeah. Anyway, if you are holding this, like you could rotate. These are methyl groups right here, CH3, and they could rotate around each other. But when you put a double bond there, all of a sudden it won't move anymore. Do you remember messing with it like that? Having a double bond totally changes the ability of these things to move in space. Okay, and that actually is important when we talk about isomers, and structure determines function, and bio is super important. So anyway, I will just send this to you. You don't read this, please. The most frequent binding partners of carbon are hydrogen, oxygen, and nitrogen, because those also want to form covalent bonds. So in bio, you're going to a lot of times see, like when we talk about these organic compounds, they're going to be made of carbon, which will be covalently bonded to either hydrogens, nitrogens, or oxygens, or other carbons. So hydrogen will form one bond because it needs one more electron in its valence shell here. Oxygen will form two covalent bonds because it just wants two more. Nitrogen can form 3 bonds, and then carbon, like I said, will form 4. So, make sense? That makes sense. So a hydrocarbon, like I said a minute ago, is a molecule made up of just hydrogens and carbons. All of the bonds are very nonpolar, so these things will not associate with water. Hydrocarbons are important components of biological molecules like fat. This is a diagram of a triglyceride or a fat molecule. And all these, the black would be analogous to the carbon skeleton, and the blue spheres are the hydrogens. The gray spheres, sorry. So this is 1, 2, 3 hydrocarbon tails. Each fat molecule is made up of 3 hydrocarbon chains. They're called fatty acids. Anyway, these are super nonpolar. Okay, all of this yellow area is completely nonpolar. Anyway, stores a lot of energy. We'll talk about fats later on. So hydrocarbons are important components of fats. Hydrocarbons are also important components of phospholipids, the things that make up your cell membrane. And this right here, the red things, are phospholipids or hydrocarbon tails. One phospholipids. You know what I know by one? Okay. This is all chapter 5 stuff. But anyways, anything that has a hydrocarbon is going to be nonpolar. And most lipids have hydrocarbon components, which is why they're all at least partially nonpolar. So they won't associate with water. They're very stable. Most hydrocarbons by themselves, just hydrogens and carbons, are gases at room temperature, like methane gas, ethane gas, butane. If you took chemistry 1, did you learn the basic naming stuff and those things? Maybe this was fine. I don't know. You might have. Things like a fat. Okay, there's other stuff in here besides carbons and hydrogens. There's oxygens over here. In a fat, we have 3 fatty acids bonded to a glycerol. So this isn't a total hydrocarbon, just these tails are. So anyway, and they store a lot of energy, a huge amount of energy, which is why we use hydrocarbons when we like in our gasoline. You burn the fuels we burn. Propane tanks, if you ever have a gas grill, you know, it's a propane tank, that's a hydrocarbon. Lots of energy when you burn it. So. Hydrocarbons can grow and they can form ring structures. So this is just showing these are what we call straight chain hydrocarbons. This is a branched chain hydrocarbon. See how it's coming off this way? And these are ring structures over here. You're going to see ring structures in biological compounds and steroids. All right. And notice in the hydrocarbons, you can also have the presence of double bonds. So these are all total hydrocarbons, but like I said, these are kind of the skeletons of biological molecules, what they're made of. So basically, how do you make biological molecules like the carbohydrates and all that stuff? Is by taking a carbon skeleton and adding those functional groups that you learned about to them. We'll talk about those in just a minute. Remember, you need to know all those for Friday. There's a lot of, I was never a flashcard person, however, but there's a lot of those flashcard things online. If those help you, if they do or not, I've never helped me. The way I studied was I rewrote my notes. I think I've told you that before. I don't know why, that's what worked for me. It's the only thing that worked for me. Like literally rewriting all my notes and reorganizing them. So, okay. So this right here is a molecule of urea. Urea is what we secrete our nitrogenous waste in our urine. Nitrogenous waste are super toxic, and our kidneys. You know where your kidneys are? Yeah, you have 2 of them. They're like right here. You ever look right here? Well, you should have 2. Some of you might not. You don't know about it. But you should have 2, and they're right in the back, and they clean your blood. So nitrogenous waste are a product of metabolism that's always being formed in your body. And one of the only organs in your body that has a continuous blood supply, like it's never ever cut off, is your brain and your kidneys. The blood supply is always on your kidneys because you clean your blood. people that have kidney failure have to do what? Yeah, so they basically take the blood out, clean it, and put the blood back in. It's a really painful, like not fun thing to do. And if someone's on dialysis, they're online for a kidney, like, and they'll want to get it sooner rather than later. Dialysis is not fun. Okay. Yeah, it's not fun. They don't take it out all at once, obviously, but it's like. Anyway, but urea, that's what you pee out. This is what the yellow stuff that you pee out. I tried it this way. So anyway. So let's talk about isomers. Isomers have the same formula, but they have different arrangements of the atoms in space. They have different shapes. And remember, in biology, structure determines function. So if someone has the same like molecular formula that has very different shapes, well, it's going to function completely differently in an organism. All of these up here have 6 carbons. The carbons are the gray dots. All of them. Now they're going to have different numbers of hydrogens, and notice their shapes are completely different. So these all 3 would have totally different functions in an organism. Or only one might work and the others don't do anything at all. You learn about isomers in chemistry?
Yeah.
My favorite isomer is an enantiomer. I have a favorite isomer. Okay, so the 3 types are structural, geometric, and things called enantiomers, which are my favorite. So structural isomers, we'll talk about first. Structural isomers, they have different, what we call covalent arrangements of their atoms in space. So let me show you something, like compare this with something called geometric. Okay, so let's go back here. So notice these 6 carbons are all in a line, and each one is either bonded to another carbon or 2 hydrogens. You see that? Here, we still have 5 carbons, but they're not in a straight line. They're branched. You see that? This is called pentane. This is called 2-methylbutane. Like it's a totally different compound. We still have the same number of atoms, but it has to do with the way the shape is. So structural isomers, their structures are just very different. Anyway, okay, geometric isomers, they have the same, so these what we call have different covalent partnerships. Does that make sense? So let's see here, the 3rd carbon here, you start on one end, 1, 2, 3, the 3rd carbon is bonded to 2 other carbons and 2 hydrogens. You see that? But the 3rd carbon here is bonded to 1 carbon and 3 hydrogens. Different covalent partnerships. See that? That's what these are. These have the same covalent partnerships. This carbon is bonded to another carbon and an X and an H. This carbon is bonded to another carbon, an X and an H. The same covalent partnerships. See that? But the arrangement in space is different. Remember how I told you when you add a double bond, all of a sudden these things can't move anymore? It's stuck in that particular conformation. These groups can't rotate anymore around each other because of that double bond. So geometric isomers are when you have this particular situation, but in this case we're talking about these functional groups X, and this particular isomer are on the same side of the molecule. See that? We call that the cis isomer. They're on the same side. Whereas here, these functional groups are on opposite sides. We call this the trans isomer. Now, in reality, that doesn't look like it's a huge difference. But what if these 2 groups are super polar? Okay? All of a sudden, this molecule now is kind of antipathic. We have a polar side and a non-polar side. But here, if the polar groups are on opposite sides, it makes the whole thing more polar. Does that make sense? So it's going to behave really differently in an organism. So it might not seem that big of a difference, but actually, biologically, it can be a huge difference. Make sense? Now my favorite ones are enantiomers. Do me a favor, look at your hands. Look at them. They look the same, right? Try to put them on top of each other so that they're like right on top of each other, exactly the same. Not exactly the same. They have to be facing the same way. How Meg try to make them? Do they go? They don't. These are what we call mirror images of each other. They are not the same in space. If you put them both facing up, you see how they're just not the same, right? The sun's coming off their own side or one side. They're not the same. So these are called enantiomers, mirror images of each other. They might look the same, but no matter how you try to do it, they're not the same. So they're going to be, like in an organism, if you're talking about a molecule like this, it's going to behave completely differently. So let's look at this molecule on the board. It's easier to kind of look at with your hands, but the central carbon is that C in the middle. And then let's look at its partnerships. So here we have a carboxyl group up here, COH. And then, so that's the same on both of them, but let's look down here. So over here we have a hydrogen and a methyl group and an amine group. So let's rotate in your head. Rotate this over here so that the hydrogen's on the same side. Rotate this at the bottom. See that? You rotated it. So the hydrogen's on the same side. Hydrogen went over here. Where did this go? It goes right here. Right? And then that NH2 group was right here. Right? You did that rotation? So is it the same as this? No matter how you try to rotate this, you can't make it match this. Because they're enantiomers, or mirror image isomers of each other. Make sense? Might not look like a big deal. You might be like, what is the big deal? It's a huge, huge difference. So in biology, because structure affects function, even a small difference like a mirror image isomer of each other will have a huge situation. So amino acids, there's 20 of them. They're written down there somewhere. But anyways, amino acid alanine. Usually when we talk about these stereoisomers, there's an L and a D version. L stands for left, D stands for right. I don't know why they call D for right, unless there's some sort of Latin word. I don't know. If it's a Latin thing, I don't know. Just for I'm goat, sure I think it's right, but no. Or drop. It's drop, right? Oh, maybe that's why. Josh is left, right? Are you a friend?
Yeah, it's just been too long.
No more friends for you? I think the new lady's like from France. I think she's from France. Like she's legit French. But there's two versions, like the left and the right. That might be it. The left and the right version. So a lot of times in an organism, so the L version is what's used, but a lot of times both are synthesized. They're both made, but only one version is what we call the biologically active version. The other one's just not used. In medication, so usually the L version is the active version and the D version is not. So down here, this particular medication is called dopamine, or L-Dopa. Dopamine is a medication used to treat Parkinson's disease. And people that have Parkinson's disease don't produce dopamine anymore. I'll move in just a second. And they slowly lose motor control. So they have tremors and they shake a lot and all that. So they can take autoimmune disorder, you can't really do anything for it, but you can take this medication and slow the progress. L-Dopa is what they take. D-Dopa just doesn't hurt them and it doesn't work. If you are a pharmacist or a person in research that's trying to create medication for treatment, you have to understand the chemistry behind the medications you're working with to understand that there are different versions of these, and then ones might work and ones might not. And you really have to understand it because of another. Sometimes the version that doesn't work can actually be really bad. Have you ever heard? Pause. There's, I think, this chart's in your book, right? So different medicines, one works, one doesn't. Thalidomide was a medication used a long time ago, I think in the 60s and 50s, for morning sickness, like the nausea, and it made them not feel that way anymore. Like if whenever someday after you're married and you have enough money and you can support yourself, and you decide you want children, because not everyone does, okay. But if you're a girl, you might have morning sickness. And some people it's really bad. I think Miss Glenn was literally physically ill when she was pregnant last time for 3, 4 months, like throwing up. I just felt bad, but I never actually threw up, so it wasn't so bad for me. But, so some people, it can be like terrible. And so you would want to take something to make you feel better. So they gave them thalidomide and made them not feel bad. But their children came out with super severe birth defects. And you can see on these children, so their arms didn't form. See that? It's like they had a hand, but they actually had no bones in here. So their hand was attached to their trunk. And it's because the thalidomide medication prevented an important part of development and caused them these birth defects. Really?
Yeah, like there's somebody with a birthday or something.
Oh, yeah. I heard about that the last time she was pregnant.
Yeah, she got it all clean.
Yeah. Ooh. I don't want to get pregnant ever again, but it's not fun. But I was never that sick. Anyway, these almost look the same. These are the two enantiomers of thalidomide. But see, what it is that the active version was fine, but the stereoisomer of thalidomide, which is produced naturally during metabolism, and when I say metabolism, your body, when you take the medication, your body breaks it down eventually, and then you excrete it like through your urine usually. So during that metabolic process, the D version or the other version of this thalidomide was produced, and it caused all these problems. I have a video to show you on this.
Afternoon. Federal announcement. Every doctor, every hospital, every nurse has been notified. Every woman in this country must be aware that it's most important that they check their medicine cabinets, that they do not take this drug.
In the early 1960s, no drug struck more fear into the hearts of pregnant women.
One of the most horrifying episodes in medical history
Than thalidomide. It changed our relationship with the drugs we use.
One reason U.S. drug laws are so strict, thalidomide.
And became an example of what many saw as corporate greed at its worst.
British thalidomide children so far have not received any compensation from the rich company that made the drug which crippled them so brutally.
But this dark chapter is only part of thalidomide's enigmatic story, one that continues to reverberate today.
I had used up every other alternative when I took thalidomide.
In 1960, a new wonder drug was slated to arrive on American shores, a sedative that was said to also treat a range of other ills.
Hypnotic, as the doctors call it, that was the answer to a prayer. Its generic name was thalidomide. The hallmark defining quality of thalidomide was its safety. So safe that in Germany there was no prescription needed.
The German company that developed thalidomide, Chemie Grunenthal, claimed that even pregnant women could take it.
The drug company had handed out samples of this drug all over the place, starting with employees of its own company. On Christmas Day in 1956, a baby girl was born in Germany without ears. And she was the daughter of an employee of the drug company, Grunenthal.
No immediate connection was made to thalidomide, which soon sold nearly as well as aspirin in some European countries.
We received it in quantities like 1,000 pills. There was tremendous pressure all over the world to get this wonderful new drug on the market.
They had 2 million tablets ready to go the moment the FDA approved the drug, which was almost a foregone conclusion, until one doctor came along and began working at the FDA.
It just so happened that my first application was for the drug thalidomide. I got this because I was new and they thought I should have an easy one to start on.
But Dr. Kelsey was uneasy with what she saw as the lack of rigorous scientific studies and the slipshod presentation of safety data provided by Grunenthal and William S. Merrill, the U.S. distributor of the drug.
The best thing that could be said about thalidomide at the time was simply that you could not kill a rat, no matter how much thalidomide the rat ate.
With thalidomide being prescribed for morning sickness in other countries, Kelsey became particularly concerned with what effect it might have on a developing fetus. In June of 1961, an article appeared promoting its safety during late pregnancy.
It was allegedly written by a Dr. Ray Olson, but in fact, the article was written by the medical director of the drug company.
About 6 months later, long-ignored evidence became public in Germany, linking thalidomide to a rash of birth defects. Although hundreds of thousands of pre-market samples had been provided to American doctors, Dr. Kelsey's stubborn delay of the drug's approval for more than a year had prevented a similar scale of tragedy from unfolding in the United States.
Dr. Kelsey was absolutely a unique hero in American history.
But thalidomide's reach continued to be felt across the rest of the world, including in Trinidad and Tobago, where Giselle Cole was born.
When I came along, I'm a firstborn, and we were a young married couple. I mean, I was never unloved or not wanted or anything like that. But I would be foolish to think that it was easy for them. My disability, the official term is focomelia, coming from the Greek meaning shorter arms or flipper-like. I think people always expect that I would have been angry. And I'm certainly not angry and never have been.
Long discussed but seldom implemented, major regulatory reforms were finally forced on the pharmaceutical industry following the thalidomide scandal.
For some time, President Kennedy has tried to get Congress to approve new controls, but without much success. Now, with the thalidomide scare, most of the opposition has melted.
Largely the same FDA guidelines that we live under today were created in immediate wake.
These regulations were too late for thalidomide's thousands of surviving victims across the world, who soon became the story.
Philippa Bradbourne is one example. Her mother rejected her.
10-year-old Carl Davies leads a relatively normal life for a boy without arms.
Another young mother, her husband, her sister, and her doctor are charged with the mercy killing of her deformed infant.
I'm one of the lucky ones, in that my parents were adamant that I was their daughter, and their daughter first before anything else, and it was treated as such. Many were put in homes because they just didn't know what to do. Some families battled with doctors to have amputation of fingers and toes and whatnot to accommodate these prosthetics. Many families were broken irrevocably.
Instead of quickly settling, the drug companies dug in, with Grunenthal originally arguing that the children's deformities were caused by everything from nuclear fallout to botched home abortions, anything but thalidomide.
It was a very long and difficult process.
Most cases were eventually settled, but litigation continues, with some survivors saying the original settlements cannot cover the cost of their specialized care. Grunenthal didn't apologize to its victims until 2012, 50 years after the tragedy unfolded.
They issued a statement saying that it's taken them 50 years to come forward to this stage because they were shocked. They don't have a right to be shocked. The shock doesn't belong to them.
Despite all that thalidomide's victims endured over the decades, they could long take solace in one simple fact.
Thalidomide is now banned everywhere.
They now ban thalidomide.
The drug was banned in 1962. And I would have liked to have seen it never used again.
There's tremendous amount of luck in science. It's almost like an Easter egg hunt.
In 1992, while conducting research into macular degeneration, ophthalmologist Robert D'Amato began his own hunt. This one to find a pill that might restrict blood vessel growth.
I started searching for drugs that cause some sort of damage to a fetus, a birth defect, and at the top of this list was thalidomide.
Although thalidomide had been banned around the world, unbeknownst to many, the drug was later made available for limited humanitarian use, thanks to a chance discovery.
In 1964, a doctor in Jerusalem was taking care of a patient who had such severe leprosy that he was in pain and couldn't sleep. He decided to treat the patient with thalidomide to see if it would help with the sleep. and we began to notice that there was also improvements generally in the leprosy.
After much trial and error, Dr. D'Amato finally had a similar breakthrough when he demonstrated that thalidomide could starve blood flow to cells, a discovery that held the potential for treating cancerous tumors.
The people that really understood the results were excited because we had a tool that we didn't have before. But the knee-jerk reaction was, this is a dangerous drug, no one ever want to use it. There was thousands of victims that still remain. There had been a promise that thalidomide would never be developed again.
We had to make a decision what position we were going to take. Many would have thought and expected that we would have been screaming, no, 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 no.
But following a surge of reports about promising studies, that's not what happened.
We are nervous, we are of course frightened and dismayed, but we realize that this is not a perfect world.
Thalidomide is back. The Food and Drug Administration says thalidomide will be the most restricted drug ever distributed.
Female patients taking the drug will even have to submit to pregnancy tests.
Since then, thalidomide has gone on to combat a surprising variety of diseases, from tuberculosis and Crohn's disease to multiple sclerosis and symptoms of HIV and AIDS. And D'Amato's hunch about the drug's effect on cancerous tumors paid off in a groundbreaking treatment for certain types of the disease.
Thalidomide and its derivatives have become the primary treatment for multiple myeloma.
It's a very fine line that I walk, and many of us walk, when we think about thalidomide in today's world. Clearly, you can see what it has done to the thousands of us who are still on this earth. But if it provides some kind of assistance, help, relief of suffering, then I cannot, in good conscience, be opposed.
Brenner, who saw thalidomide's curative powers firsthand after being prescribed it for a deadly skin disease, says that the drug's circuitous history tells us a great deal about the accidental nature of drug discovery.
It's kind of surprising, I think, to most people to learn that drugs are, in fact, developed and then become, as they're called, a drug in search of a disease.
When you have a drug that has a powerful activity, whether it's negative or not, that powerful activity could be used for something good if you could figure out what that was.
But for all its benefits, the scientific rehabilitation of thalidomide has come with a heavy cost.
We were told this could never happen again.
In Brazil, where the drug is used extensively to treat leprosy, researchers report that thalidomide children continue to be born, as many as 100 since 2005 by one recent estimate.
50 years down the road, I would like to think that there's no such thing as thalidomide. That we have created something, that we have developed something that would allow us to bury thalidomide. Literally. Destroy it. Get rid of it. So that there are no further discussions other than another history.
Interesting, right? Anyways, there's a lot of medications that are discovered on accident. You know, Viagra is like, Viagra. The way Viagra was originally developed was to treat people that had heart problems. And all these men were taking it, but they were like having to talk about their side effects, and they kept saying they all were having the same side effects. And then the drug company was like, this is more marketable. Maybe we should use it for this reason instead, and that's what it's used for now. Anyway. Yeah, it does. It's like a yes, but it can still be prescribed for that. But like I said, the more marketable reason now is for erectile dysfunction. Thalidomide? No. And I think because of my age, it's not a medicine that my doctor would ever want to use on me right now. Because I could still potentially have children. The medicine I am on, the one I started last year, like on the pill or on the bottle, it's like I'm not supposed to take it if I get pregnant. So I take birth control to try to, it's not, I think if I did, I just have to stop it right away. And even the other medicine, the shots I give myself, like my OBGYN wouldn't let me do those while I was pregnant with the twins either. But, and it's because they weren't sure how it would affect, but it could cross the placenta. They know that. So yeah, there's, like they said in there, something like thalidomide, they would even make you do like pregnancy tests. I think Accutane, did I ever take that? What do they do? Then there's something weird with that. Yeah, like Accutane, the medicine for acne, I think girls would have to take pregnancy tests with that too, because that's really bad. Yeah. So, okay, I can go through these pretty fast, so we're good. Now we got 59, right? Okay. Okay, so a lot of times these functional groups, and like I said, there's a chart in your book. Hopefully you look at it, you need to study that whole chart. So by adding a functional group, you totally change the chemistry of a compound. Most of those functional groups have the tendency to increase polarity, make them more hydrophilic. The only functional group that makes the molecule less hydrophilic is the methyl group, or that makes the molecule. We'll talk about it. So ethane was a hydrocarbon. Here it is. C2H6, and if you add a hydroxyl group, which is the OL, here, all of a sudden you have ethanol. Ethanol is what is found in, beer and stuff. It's the alcohol you drink. But it all of a sudden changed a molecule that's completely non-polar to a polar molecule, at least over here. And all of a sudden the gas at room temperature, ethanol is liquid at room temperature. Changes its chemistry, chemical properties. There you go. Like you're like Heineken, I don't drink beer, I use the disgusting, but there you go. I like wine. So let's talk about them. So these functional groups are these part of organic molecules that are important in giving it the biological properties. A lot of times they're involved in reactions. And these are all the ones you need to know. I'll go over each one individually. So down here we have, what are these called in general?
Hormones.
What kind of hormones? Not all hormones are these. Steroids, whoever said it, yes. Influenza hormone, influenza out of a protein. By the way, not all hormones are steroids, but these are steroid hormones. This is estrogen, which is found in females, and this is testosterone, which isn't found in males. Mostly they're the same. They have this word ring structure, which all steroids do, and has this OH and methyl group up here. Testosterone has a methyl group here, and has this ketone here, and then estrogen has the hydroxyl group. These two differences make a huge difference in the functioning of those molecules biologically or in an organism. Testosterone is what makes, like, for example, male lions grow their really big manes, and the more testosterone they have, the darker their manes are. The estrogen and the lack of testosterone, the females, won't do all that. They're not aggressive, blah, blah, blah. The functional groups are super important. And then you notice, you're going to see a lot of bird videos in the past because birds are really crazy and have these really weird, like, mating rituals. And you see a lot of what we call sexual dimorphism in birds. Do you know what that means, dimorphism? Morph is like a form. It's different. Yeah, and di, B2. So sexual dimorphism is different from the sexes. And you see it some in like us. I mean, obviously males are different from females, but if you cut our hair the same way and everything like that, it might be kind of hard to tell the difference sometimes unless you're just going naked. But in birds, you really can tell the difference a lot of times. So like cardinals, for example, the female cardinals are brown. They have a little bit of red, but this is the female, and the males are the pretty red ones. In most bird species, the males are the ones that are colorful. They're the ones that they sing the most. They do these crazy weird dances and mating rituals. The females are like boring. Sometimes the females are a little bit bigger, and but they pick their males. They pick based on the male's color and how he acts. It's actually really weird. So over here, same thing. The male's the pretty one, and here's the female. She's boring. So she picked him. So he's doing this to really just impress her. He's like, look at me, I have this friend. Seriously. So, but you see sexual dimorphism in other species as well. Fish, some fish species have it. So like bison, The males are going to have the horns, same thing with like the deer. The males have this big racks versus females don't. But anyway, so this all has to do, and here we have the males that's really big on the females. But it's all based on the hormone differences produce these mores. All right, so the first group we'll talk about is the hydroxyl group. It's OH. Now I'm going to show you something really fast. Whenever you have something written like this, OH, okay, first of all, this line is indicating that this oxygen is covalently bonded to a carbon skeleton over here somewhere. Like this, see that? Okay? Now this OH is not just like it's actually an O that's covalently bonded to an H. That's actually what's going on there. They just write it OH for some reason. But there's a covalent bond in there. They just don't ever draw it. Just want you to understand that. So anything that has a hydroxyl group, we can call an alcohol. Hydroxyl groups are super common in carbohydrates. There's lots of them. Ethanol is one, but ethanol is not really biologically important. But increases, what you need to know is it increases the hydrophilicity, makes them more hydrophilic. Because that OH is a very polar covalent bond right there. So you can have partial charges. Right? And unfortunately, there's not a whole lot to do except for memorize this stuff. All right, this is called a carbonyl group. And it's anywhere there's a C double bonded to an O. Carbonyl groups can be in different locations within a molecule. Okay, so your book has the examples. Carbonyl groups are also common in carbohydrates, by the way. So whenever there's a carbonyl group at the end of a molecule, so if you look down here, see we have our carbon skeleton, and here's a C double bond O and then to an H. And that's the end of the molecule. There's no more carbon skeleton over here. Okay, whenever it's at the end of the molecule, we call it an aldehyde. And whenever it's in the middle of a carbon skeleton, like here, okay, it's called a ketone. And so these are common in carbohydrates. All right.
Wait, what's the still other one?
Well, what?
I was just the one.
The ketone, yeah. What? No, it does not matter. She asked if it mattered what end the aldehyde is on, and it doesn't. See what page this is. Page 63, y'all, in your book, that chart. Page 63. Like, if you're going to study anything in that chapter in terms of diagrams, you really need to know that. The other diagrams, not so much, but that one, yes. So. Okay. Carboxyl group, COH. So what's going on here, a lot of times they write it like this, but what this actually means is this. A carbon is double bonded to an O. So similar to this, but the difference here is that instead of just the H, it's an OH. You see that? Okay, this is called a carboxyl group. These are always at the end of molecules. Anything that has a carboxyl group is called a carboxylic acid. It can function as an acid because it can ionize in solution. That H can go away, and it can do this. Okay? Carboxylic acids are in amino acids, which make up proteins, and they help to make fatty acids, which make up fat. And there's fatty acid tails on phospholipids. Definitely makes it more hydrophilic. Yes. All of these functional groups are, really, except for the sulfhydryl and except for the methyl. But yes, hydroxyl groups are because of the O and the H bond. It's a really polar covalent bond. All right, these are two pictures of fatty acids. See the carboxyl group at the end? I just want to show you that. Oh, amino groups, NH2 is a nitrogen bonded to 2 hydrogens. These are found on amino acids. Amino acids have both. I'm going to go back, hold on. And this is the structure of amino acids. Do you remember this in Bio 1? Okay. Amino acids have both carboxyl groups and amino groups. Okay. So we'll talk about that in chapter 5. But it's in bonded to 2 H's. They can also ionize in solution, but they have a tendency to accept hydrogen ions and function as a base, interestingly enough. But they also increase hydrophilicity, not just because it ionizes, but also nitrogen and hydrogen, those bonds are pretty polar. There's really not much except for just memorizing this stuff. All right, sulfhydryl groups. Whenever there's a sulfur bonded to a hydrogen, anything that has a sulfhydryl group is called a thiol. The reason these are biologically important is because they help to stabilize protein structure. Do you remember in bio, I always say this, I'm sorry. And Nick's probably just like, no. It's like all semester, just your answer, I know is no. But tertiary structure, he didn't ever take bio 1. Long story, just don't worry about it. He's fine. Primary, secondary, tertiary, quaternary, y'all. Yes-ish. Yeah. Anyway, the tertiary structure of protein, sulfhydryl groups are important in stabilizing protein structure. If you don't know it, sorry, we'll talk about it in chapter 5. But they have it creates things called disulfide bridges, which is what's showing here. This bond between the S and the other S. A covalent bond. People that have super curly hair, like I have like wavy hair. It used to be curly when I was like, the more curls you have, the more sulfhydryl bonds there are in your hair. If you ever get a perm, like a curly perm, they put chemicals in there to make more of these disulfide bridges between your hair. You straighten your hair, get like a straightening thing, they put chemicals in there to break those bonds. So I'll talk more about that in chapter 5 as well. But sulfhydryl groups, there you go. Yeah, so it's just the disulfide bridge there. It's in protein. Phosphate groups, you have seen this before when you talked about ATP. I know you did in Bio 1, but a phosphate group is super polar because it can ionize twice and have really like largely negative charges. This is the way it looks like bonded to a molecule. Okay, so it's like you're like, where's the carbon? Carbon skeleton will be where these red dots are over here. Phosphate groups are an important part of ATP and things called GTP and TTP and CTP, which you probably didn't learn about before. They're very reactive, and these groups function sort of an energy transfer. We'll talk about how that actually works later on. Phospholipids have a giant phosphate head or phosphate group in their polar head. And then a methyl group. A methyl group is just CH3. Methyl groups are important generally because they Did you learn, do you know what to express DNA? What does that mean? DNA is expressed. Yeah, it kind of it shows up. It's like it's being transcribed and translated. It actually makes the protein. Yeah. So gene expression, okay. So, you know, every cell in your body like has the same DNA. The cells that make up your skin are the same DNA that the cells make up your heart. The same exact DNA. And how can they be so different? Anybody know? Yeah, has to do with DNA expression. Not all the genes are expressed all the time in every cell of your body. So how do you turn off genes? With this. Genes are turned off in your cells through methylation. They're highly methylated, or methyl groups. These CH3 groups are added to your DNA. And when there's a bunch of methyl groups added to your DNA, that part of your DNA can't be read, basically, transcribed and translated. It's not expressed. How that happens, we talk about in like chapter 16 and on. 17, yeah. But that's that.
You sometimes keep methyl groups and like you DNA what happens.
Well, it'll be expressed. I mean,
All of it, like,
it's more complicated than that. There's a lot of ways that gene expression is regulated, and methylation is not the only. I mean, there's complicated, not necessarily. You can change your methylation by the foods that you eat and by the chemical groups. Your epigenome. Anyway, yeah. Sorry.
why is methyl, why are they not hydrophilic?
Because all the bonds in there, the C and H bonds, they're nonpolar. They have similar electronegativity, so they share the electrons equally. Yep. So methyl groups can also be found on things like your steroid hormones and stuff to affect their functionality. But yeah. Now I want to point out something real fast. The end of your chapter talks about ATP. End of chapter 4, like specifically, like there's a blurb about it. If you're one of these people that's like super obsessed with the textbook, I'm not going to test you on that on this test. Just that little part right there. Just FYI. Okay. Good. Yeah. Okay. Well, if I ask you about it, it'll be in terms of like that, but that's all. Like this one talks about there's 3 phosphate groups and talks about ATP going to ADP and all that. I'm not going to talk about that right now, so I won't test you on it right now. We still have a little bit, so let me talk to you for a minute. I'm going to show you something. I'm not going to move on yet. Does anybody have test anxiety yet? Not really? Okay. I know.
Oh, okay.
I'm doing this on purpose.
Oh, okay.
This is from 2010. No, this is old. I want to show you something. Now, whenever I took tests, like I would have a lot of test anxiety because I didn't know how detailed the questions would be, the level, all this stuff. These tests are hard. But I'm telling you, those review packets I emailed you, I've said it like so many times now, do those questions. The style of those questions, the difficulty is similar because it's the same test bank. These are some of the types of questions that I like put on an old test like 7 years ago. You might actually see these tomorrow because some of the questions are the same, but things like this, okay? And I'm going to tell you these questions right now, the ones I put on your test are more for me knowing that you know the content. When we start getting closer to the AP exam, the questions are going to change a little bit. They're going to be more like, more analytical. A lot of these are more like rote memorization or like, do you actually know the stuff? Does that make sense? For every AP class you take, there's something called an Acorn book. Have you heard about these? You've never heard of an Acorn book? Oh my God. Okay. This is going to help you with all of your, the College Board puts these out. Watch. Watch. I'll just show you AP Bio, but I'm telling you, well, AP Psych, just show you this. Now, the Acorn book, the reason it's called an Acorn book is the course description for the College Board makes these. This is called Acorn because in the corner it's an acorn. See that? For every AP course you have, there's one of these books. It talks about everything you need to know. The useful thing about these books is there's practice questions at the end of all of them, just like on the AP exams. All right. So there's one of these for AP Bio as well. And I'm going to show you that one real fast. I don't know. Yeah. I want to show you something just real fast. Like this is an actual AP, more like what the questions are now. There's usually like a cat of words and like diagrams, and then the question related to that on the AP exam. So anyways, I just want to point that out. But look, what are trace elements? And you have to be able to identify them. Some of these questions are going to be like, which one of these is false? I hate questions like that. And I'm sure you do too, but there will be some like that. That means there's only, the rest of them are right except for like one. Or some of them, which one of these is correct, and they're all wrong except for one. There's questions like that. There's not a ton of diagrams on this stuff. I want to show you this. There will be some like this on Friday. These aren't these questions necessarily, but you have to identify functional groups just by looking at a picture. Okay? And the question like, which one has a hydroxyl group? Okay? So like there's a hydroxyl group. Which one has a carbonyl group? Blah, 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 blah. Okay? So that's the kind of the simple structures, not complicated big ones yet. I want to prepare you. Some of you start studying it tonight, okay? Don't be stressed, but you know, and then yeah. Yeah. Oh yeah. You can.
Number 2 will have to be valid. Answer C.
I don't know. Yeah. That's tomorrow. Tomorrow, y'all are going tomorrow. You don't know. I can tell you have to be here all the time. You can't leave like all of it. Okay. You don't. There's a week in January, February. We got an email like somewhere in there. End of January, beginning of February. Make it work. A lot of stuff we don't like, who knows that?
One of the so like
right, yeah. I think some of the stuff is like just crazy random bio knowledge that some people really have, and we don't necessarily learn it in here or at all in high school. Thank you. Bye. If they're juniors.
I think they said it has some juniors in it.
Yeah, not sophomores. But sure, thank you. I appreciate it. Where in the world?
I'm gonna grab two more, give these two back. I'm gonna update them all.
Thank
They're
you.
they're actually really bad.
Is it really that bad?
I mean, it's like 2.1 and the current one's 2.7.
Oh, wow. Thank you. Hey. How are you?
Good, how are you?
Good.